I'm Inyoung from Seoul National University at South Korea. Today, I briefly present my recent research with poster number 791. First of all, I would like to thank ISMB for giving me the opportunity to present a poster at this great conference. The title of the research I will present today is MDTR, a knowledge-guided interpretable representation for quantifying liver toxicity at transcriptomic level. So let's start the representation. This study aimed to measure the degree of the liver toxicity in various environments. Therefore, we used drug-treated transcript data as input and proposed a multidimensional transcript level distance based on the biological knowledge. The key points of the model are that an outlier detection model was used to select the most perturbed samples, and the embedding space using the nonlinear kernel was constructed based on the most perturbed sample and biological prior knowledge. Through this, we propose a 5-dimensional liver toxicity ruler that can measure the degree of the liver toxicity. The background of this study is that drug-induced liver injury is a very important issue in drug development and patient safety. However, most delay prediction studies are conducted in binary form at the chemical level. Moreover, although it is the clinical drug response of the interest in practical, but the data on this field are lacking. So if we analyze this at the transcriptomic level, we will be able to bridge the difference between the chemical and clinical level. However, there are 3 major problems with transcript data analysis. First, transcript data is high-dimensional data with more than 2,000 genes, but the number of the data that can be analyzed is very small. Second, based on the current knowledge, toxicity signature at the transcript level is not clearly identified. Last, the level of toxicity cannot be limited to any boundary. So, this study aimed to measure the degree of the toxicity for the different environment in which the drug was treated. The perturbation signature of the links is known to be associated with cell viability, linked to the regulation of the transcript factors such as apoptosis or proliferation. In particular, toxic compounds can induce cell death signature. So, this suggesting that the potential to prediction most mechanism of action through drug-specific perturbation profile. Therefore, we assume that the toxicity signature will show distinct perturbation compared to the other, and the degree of toxicity of the sample will be measured based on the disturbed sample. Therefore, in this study, we propose a distance called MDTR. First, in the data-driven view, the most perturbed sample and the data are selected. We proposed a dual SBD based on the outlier detection model, which identified the most perturbed drug-treated samples through a first boundary that distinguishes drug effect, and a second boundary that distinguishes toxic effect. Next, in the knowledge-driven view, 5 mechanisms were selected through a research search for liver toxicity, and its mechanism was further subdivided into biological pathway using KEG pathway. Finally, for each pathway, an embedding space was constructed using the RBF kernel corresponding the gene in the pathway based on the most perturbed sample selected by dual SBDD. And the pathway level distance was measured through Mahalovin distance in the embedding space. Furthermore, the mechanism level distance was measured by integrating the pathway level distance belong to the mechanism. So we create a 5-dimensional liver toxicity ruler for a total of 5 mechanisms. In this study, liver toxicity labels of drugs were collected through two databases, DILI-LANC and LiverTox, and drug-treated transcriptomic data was collected through the LINCS database. Let me explain the main result of this study. First, to show the robustness of the proposed distance, we created distance using different sample sizes within the data and calculated the correlation between the distance created by each. As a result of self-validation, it was shown that the proposed distance was calculated consistently regardless of the sample size. Next, the MDTR distance were measured in the distribution shown on the below figure. Next, we examined the correlation between the measured distance and the dose of the drug. For this purpose, we compared it with the following three methods, and as a result, the distance of the proposed method showed the highest correlation with the dose. Lastly, we performed case study and it was shown that the MDTR has an interpretable power according to the dose of the drug and can determine the potential toxicity of the drug through the change in the distance. So, to summarize the content of this study, in this study we proposed the MDTR which measured the degree of the toxicity according to the 5 liver toxicity mechanisms in transcriptomic data. So, this is the end of the present of this study. This is our lab member and the correspondent author Professor Sung Kim and co-first author Dr. Sang-Sun Lee. Thank you for listening, and if you have any question, please contact us at the below address.